We'll start again, but I won't say what I just said again. So jaws are this big piece of vertebrate evolution, lets body size take off. Oceans are pretty much dominated in terms of most of the big predators by vertebrates, and they're jawed vertebrates after the Devonian. Okay. Now, get in here. So this is where we actually left on Thursday, right? You guys learned a bunch of different synapomorphies that unite the jawed vertebrates today, which are right here, but you've seen how those synapomorphies are spread across this gnathostome stem. Some of these features apply to all these groups. Some of these features only apply to one plus gnathostomes. Some of them really are, these three, if you remember what those three are, only do apply to jawed vertebrates. This is kind of our context for the origination of gnathostomes, of jawed vertebrates. And so that's really cool to me to put on this phylogeny. So this is a phylogeny of living, modern vertebrate biodiversity. Every single picture of an organism on this slide is a living thing you could go see in the natural world today. And so we have nodes on here that you guys are familiar with, but these synapomorphies all crowd each other up. Look how many there are. 1, 2, 3, 4, 5, 6, 7, 8 gnathostomes, if you only look at the modern. These are the same 8, 1, 2, 3, 4, 5, 6, 7, 8, spread across the fossil record. So again, these fossils let us see how body plans are built, how evolution happens over time. It's not like you don't know what comes first, bone or pelvic fins or jaws. There's an order to these things. And so you guys have already seen this now a bunch of times. Vertebrata is a monophyletic group with two sister lineages contained within it, a monophyletic cyclostomata and a monophyletic gnathostomata. And we're finally getting to those gnathostomes, those jaw mouths, jawed vertebrates today. And now I'm going to give you guys a bunch of this fish taxonomy. These are a bunch of fish words we all need to have in order to talk about fishes. And it's kind of a bunch all at once, but then we're going to be able to use these words for the whole rest of the class. So once you break up vertebrates into cyclostomes and gnathostomes, you're good. Once you break up gnathostomes, you get two more clades. Osteichthyes on one side and chondrichthyes on the other side. Ichthyes is fish, which maybe isn't a surprise to anybody. Maybe it is. Oste is bone, and chondrichthyans, that's cartilage. So you have your jawed fishes are either bony fish or cartilaginous fish. And remember, this is us only looking at modern living biodiversity. So of course, when we have fossils, it's going to get messy. But so we have gnathostomes divided into bony fish and cartilaginous fish. The osteichthyans themselves are divided into these words, which some students think are terrible, I think are fun. Sarcopterygii, with two eyes at the end, and actinopterygii. The pterygii has to do with like their wing or their like limb. It's this thing. And so actin is like there's ray-finned fishes, and sarcop is like muscle, so lobe-finned fishes. So bony fish are either ray-finned or lobe-finned. So we can make this easy for you guys. I need you to know those words, and we're going to talk in those words. But just so you can see it, if you have a vertebrate, you're either a circle mouth or you have a jaw. If you have a jaw, you're either bony or you're cartilaginous. And if you're bony, you're either a ray fin or a lobe fin. So I'm going to use Sarcopterygian, I'm going to use Sarcopterygii, I'm going to use lobe fin pretty interchangeably. And I want you guys to get comfortable with this. When I say things like fishy fish or ray-finned fish, I'm talking about actinopterygian fishes. That doesn't mean a lamprey isn't a fish, or a lumpfish isn't a fish, or a shark isn't a fish, in quotes. But ray-fins, actinopterygians, if you guys remember from our first in-person lecture, lecture 3, we talked about modern vertebrate biodiversity. Nearly half of all living vertebrates are actinopterygian ray-finned fishes in terms of the species that are alive today. So there they are. This provides some structure for us in the living world. Once we have the fossil record, you guys are going to see why the Devonian is crazy. It's really the Silurian too, but the Devonian is where we see a lot of the really great fossils for all of these groups. Okay, so brace yourselves. There is what you've seen so far, right? A time-calibrated Paleozoic stratigraphic phylogeny of vertebrates. Everything except for that lineage called Gnathostomata is a jawless vertebrate with a cartilaginous like endoskeleton. Some of them have cool bony armor. Here's what we're going to do today. Sorry. Bust those jawed vertebrates up. And so I'll give you a second to kind of take that in. And so that same phylogeny you just saw with cartilaginous fish and bony fish, with lobe-finned bony fish and ray-finned bony fish, that's all still contained here on this slide. But now we've added time and a bunch of cool fossil lineages so we can talk about the actual evolution of these different groups. So we're going to get into all of these. You're not going to get them all at once and then I just move on again. We're going to be in this tree for the whole rest of class. So there's our vertebrata node. There's our circle mouth node of today's crown cyclostomes. So lamprey and hagfish, and then here's these two lineages you've already met, conodonts and anaspids that are on that side. All are gnathostome stem. Something that gets fun in the fossil record is that when we talk about what we call things, we have to make choices. And you're gonna see this when we talk about birds, you're gonna see it when we talk about mammals. This is another fun example, gnathostomes. A lot of people would put the gnathostome node right here, and I put the word total in parentheses. This is the gnathostome total group, meaning literally this and everything past this node is where somebody's got a job. That's the gnathostome total group. I hope you guys can see the utility, maybe, of also having a gnathostome crown group. So this is the common ancestor of every living thing with a jaw, but on the fossil record tree, this is like where jaws actually show up. Does that make sense? Okay. There's your chondrichthyans, crown group chondrichthyans, and osteichthyans. And then within your osteichthyans, there's your sarcopterygians, and then that giant bar on the far right is the one literally called actinopterygii. So I've broken up the other groups. But you can see that same set of terms that's right here is also able to be put on this phylogeny. So bouncing back and forth between the modern things you can go see at the zoo and the aquarium and the fossil record is like one of my big goals here is integrating these things for you guys. Okay, now I'm replacing the magpie and the hammerhead shark with like actual Paleozoic things, because once you get things called tetrapodomorpha, you don't get birds for a real long time, but that's where they're gonna go. That's where you go. So now the artistic renderings of these animals are all time appropriate versions. So look at all these early vertebrates. Some of them are actinopterygians, some of them are sharks in the true sense, some of them are jawless fishes. There's a whole lot of Paleozoic vertebrates right there. These two groups are grades, they're paraphyletic. Some of them are closer to you than others are. And historically, they used to be thought of as like monophyletic. We didn't really use that word back then, but groups where they all go together. Placoderms we're going to talk about today. These are some of the really crazy armored jawed vertebrates. Some of the most famous Paleozoic animals that you'll see in like a cartoon or something are placoderms. And then there's these weird sharky things called acanthodians. I put that on there because those are grades. They're still something we can talk about intelligently, but this is how they're distributed on the phylogeny in 2024. Okay, now's your chance to, I'm going to give you to talk to your neighbor. I've grabbed the Devonian and pulled it all the way across the whole page. So now the Devonian time is very easily highlighted. Talk to your neighbors for a few minutes here. Can you see why the Devonian is called the age of fishes? Okay, go ahead. Get detailed about that. Absolutely. Yeah. Doing a lot of and you've got several class in the writers. Bonus saying that yeah, lots of other things. Well, sure. Yeah, one. However, you have to get which were these? I think you're right. There were early. Yeah. Yeah, they were the tetrapodomorpha about it. Those are they start out as some book and All right, can I get some volunteers? What are some things you guys are talking about or observing when you see all this all at once? I'm hearing good stuff. Yeah, certainly.
It would like be easier to fossilize than the biggest fishes.
How come?
They have teeth.
Oh, teeth? Yeah, a lot of them have teeth. Not all of them have teeth, but yeah, maybe there's teeth, so teeth make a better fossil record. So maybe that's part of the story of how we track them down. Okay, what else?
Almost all of the nature groups are present.
When? You mean in the Devonian?
In Devonian, yeah.
Yeah, right. I mean, we can talk about lobe-finned fishes, and you guys might think, oh, there's coelacanths and lungfish and tetrapods. So we are the tetrapods, right? Tetrapodomorph fishes are fishes that are closer to us than they are to any other living fish. Something that I think is very cool about the Devonian is like coelacanths and lungfishes and our clade that you and I are in, they're not like necessarily older or younger than each other. And that's true here. The oldest like members of this lineage, which really is going to be called elasmobranchs later, but don't worry about that right now. I'm calling it sharks right here. Like you guys have probably heard a million times how old sharks are, but like there's tetrapodomorph fish fossils older than any elasmobranch fossil. So there's not really like a linearity to this. I always, well, what else? What other things people notice before I just play? Is it too many fish? That's kind of the story of vertebrates. It's a lot of fish. Give me something else that some group was talking about. I heard a lot of good stuff.
Notice how you have all the stem stones that are basically the same time as the crown group, but then they all go extinct by the Devonian.
Yeah. So a lot of these groups do seem to go extinct, like they don't make it out of the Devonian or somewhere in the Devonian. That's interesting, right? You have these lineages like conodonts, which go right through. You have some of these lineages, like this is an acanthodian lineage, but we think it's actually more closely related to today's cartilaginous fish, and it survives right through. So there's differences there. Something I noticed, and when I first taught this class ever, was the first time I ever actually was like, oh wow, and like looked into like all the actual fossils that went back as far as they could go. And like even some of these clades of jawless fishes like aren't older really than a lot of the splits we know between all these groups. So I've shown you all these jawless fishes, and now we're talking about jawed fishes, so it feels like this happens, then this happens, but like timing wise, like some of these clades, like Galeaspids and Osteostracans with this little armor, their fossil record isn't actually like much older at all than what we literally have jawed vertebrates for. So there's like this comes first and then these replace them. Like these are living happily alongside these things almost the whole time. I just think that's kind of subtle and important. I already asked you to talk about why this is the age of fishes. I'm going to ask you to dive a little deeper. You already made a couple observations. What questions does a diagram like this put on you? And you can think big about this. You don't have to be like really specific about the tree. What questions do you have when you're shown time on this, we can call it the y-axis or the left-hand side of the screen, and then all this diversity happening? What are some questions you could use this picture basically to ask? And what are those questions? Okay, talk to yourself about that, please. Or near about that.
Well, I had a question once I hear us talk about, you know, the fossil age of sharks. It's not as old as the latest one. I'll try to show. So, well, you know, we just keep saying, we ran into this situation. We had to be a little bit of like, what else, you know, first hour, we just had, you know, right? So my question was, what was part of the how do you allow cartilage fossilization as well?
What concentration is like probably all. The one who judge, so they better have tax station. Yeah. This is right. But there's a huge job. Then not have
Yeah.
All right, I'm gonna get 3-4 questions up here. What are some things you guys are wondering about? What questions do you have? How might you ask them? That kind of thing. What are you talking about right now?
I got one.
Yeah, Henry.
Yeah.
Henry's group.
How come cyclostomates and conodonts are the only jawless fish that make it past the end of the Devonian?
So
Like what happened to all those, like what happens to all the stem Gnathostomes, the little armor guys and the little armor guys in the bottom of the ocean?
this is a taxonomic word you can use. Agnathans are just jawless fishes. Obviously, there's all kinds of ways those are related to each other, but what factors allow a few agnathans to survive, like out after the Devonian, we can just say. That's an interesting question. That's something maybe worth exploring. Lampreys and hagfish are the only ones that are still alive. Cool. All right, what else? Yes, sir.
I'm wondering, like, the common ancestor of some of those groups, that we don't have fossils of, would you recognize them as looking at, like, what they eventually evolved into? Like, the common ancestor of the ray-finned fish, would you even be able to tell it apart from a float-finned fish early on?
People, is that question resonating with people? That's like a really, really, really important problem. It's like, and it is a problem. You're gonna see a couple of them on Thursday for the one node you specifically just talked about within osteichthyans. There's plenty of there's a couple Silurian osteichthyans that we have fossils of, and sometimes they go here, and sometimes they go here, and sometimes they go here, sometimes they go here, depending on who's looking at them and how they're interpreting the anatomy. It's hard. How do you recognize this fish versus that fish? When everything's fishy, superficially, right, they all look very similar. So how am I like, this is a shark, and this is a tetrapodomorph fish related to us? Interesting, right? Okay, what else? Yeah.
We were, what about the, like, ancestral total Gnathostomata groups?
Yeah.
Made them less fit for survival than the crown.
Yeah, you mean like these lineages here?
Yeah.
Like why,
What are
maybe
the
today there could be a node here called the crown because this one is still alive. So what's the deal?
Yeah, what were the characteristics?
Yeah.
That made them less successful? Or
That's interesting. I'm gonna say of most early main stones. It might look on my slide like there's a bunch of amazing success on the right and a bunch of extinction on the left. That's partly due to the fact that I'm trying to emphasize some of these clades. Within here and here and here and here and here and here, there's plenty of gray bars that go like meh, and then stop. But the total group, because there's like 2 living coelacanths today, their arrow gets to go all the way through. There's, whatever, 30-some thousand of these today. So these are different. And it looks like these are all killing it, but there's a lot of extinction here too that's just not obvious on this slide. But this is a really interesting question because it certainly seems to be the case that here's the earliest jaws and a bunch of these split. None of these ones are around anymore. So, okay. One more. These are the kind of questions I like to have you guys pondering. And anytime you ask a question like this, as we go through class, I think you're gonna get better and better at it. Thinking about what data you need to answer this, how you might answer this. And that's a kind of a nice little, like for yourself, hypothesis testing. Because then if you actually look into the literature, maybe no one bothered, maybe no one can, or maybe people have, and you were kind of on the right track. And that's how people track. Anyway, one more. Yeah, guys.
You you talk a bit about, like where exactly would the lungs show up, right?
Okay.
Lungfishes have lungs, tetrapodomers have lungs, it might be from the swim bladder, when did that stuff show up?
So you might be thinking, not all of you have this necessarily, and that's definitely okay. Prior knowledge about the biology of these animals. All these things are oxygenating their blood. I think you probably know that everybody, except for maybe this one, has gills, part of their throat, where they can like get oxygen out of water. But obviously some of these things breathe air with lungs, which are extremely different. So where's that going on? We'll talk about that on Thursday for sure. Okay, thank you guys. Thank you for these questions. These are fun things to think about. That's my last question for you. So it's fun for me because fishes as an idea obviously makes sense. It swims around in the water, it's a fish. But in terms of evolution, it's paraphyletic, right? Your vertebrae and your skull are down here. Your arms and your legs are like here and here, are here and here. So some parts of your bones are here, some parts of your bone are way up there. The stuff that you guys are sitting around using right now is spread all through this tree. Obviously, there's a lot more that makes you a mammal and a primate and stuff, but you have a lot of fishiness. Okay, so here's how we left things off. I want to talk to you guys about diversity, the fun stuff, I think. So these first 3 boxes here, there's actually many more boxes than this. I just made it 3 and chose 3 of my favorite representatives. This is just the cool biodiversity of these animals. So let's talk about placoderms. Placoderms are some of the most abundant and easy to find fish fossils, jawed fishes from the Silurian and from the Devonian. They are everywhere. They are extremely biodiverse. There's tons of species. They live in fresh water, they live in salt water, they live all over the world. They're extremely heavily ornamented. So I want to show you a few of them and then have you talk about them. So there's a nice diagram from a textbook of a bunch of different placoderms. Here's an actual fossil of one called Lunaspis. It's an early one. You can see it's got this big old head shield. There's its pectoral fins with these big spines on them. I hope you think it kind of looks sort of similar to some of these jawless fishes we've already met, like those osteostracans, with that sort of body plan of the tail behind a big armored head. But they don't all look like that. Here's one called Phimolepis. Phimolepis is very cute. The armor gets really ornamented and interesting. This is a whole clade that's very successful of placoderms called antiarchs. And these are their little eyes. They got little windows in their armor. So both eyes are way up front. And they also are bottom feeders. They have the teeniest lower jaws you can imagine. It's like a little thing that barely moves, but it is a whole lower jaw. And it's on the bottom of their face. So they're living on the bottoms there. Lunaspis is also probably an animal living on the bottom. So similar ecologies in some of these early groups to those jawless fishes you've already met. There's Brindabell-aspis, which again is one that lives mostly on the bottom, we think, because its eyes are on top of its head, nice and armored. I put that one on there because I like saying Brindabell-aspis, and I think that's a fun name. Without question, the most famous placoderm and easily maybe the most famous Paleozoic vertebrate in the water is Dunkleosteus. Has anybody heard of Dunkleosteus before? Yeah, Dunkleosteus is cool because it is so huge. The skull there, and hilariously, most of these fossils come from like Cleveland, Ohio. Northern Ohio is where these things are found the most. Is like, if you got that armor and that head, it's like not unfair to say it's like basically like this desk in terms of how big it is. So it's a big old like meter stick, could be held right there, probably within that back armor. It might be a little like this much of the meter stick in its armor. This is a really, really, really big predator. It's featured in like cartoons and paleo media all the time. So one thing you might notice about it is that it has a pretty horrific looking mouth. Depending on who's doing the study, you do get results that, especially at these like really focused pressure points up here, Dunkleosteus has one of the most like absolute power in terms of like newtons of energy bites that's ever evolved, which is really, really, really interesting and cool. I hope you can see ways that armor has gaps in it to allow for movement. So like this top of the head here, there's a gap behind the top of the head where you get to like the armor that's on the back. And so we know this animal can really like pop its mouth open really wide and then slam its mouth open really shut. It's a really interesting animal with a really interesting ecology. It has these bony lower jaws, doesn't have any teeth. The actual bones themselves are like the things that are shink, which is a little bit intense, right? There's no teeth in that mouth. So Dunkleosteus is really interesting. The more like brain rotted paleo nerds among you might know that this animal's been thought to be something like 25 feet long for a long time. There was a paper that came out maybe last year or 2 years ago, last year, that did a whole reassessment of all these different animals and how their body plans might be, because all we really have of their fossils for the most part are their armored front parts. That this big one might be like actually very stubby and short, and maybe something like, what is it, like 10 or 12 feet long. So it's basically like a big Pac-Man head with fins, which is really fun. There are smaller relatives of Dunkleosteus that we have the whole skeleton of, so this isn't some outrageous body plan. But it's interesting to think about that for this really, really big one, because the cartilaginous skeleton inside of its body, its vertebrae and stuff, don't really fossilize. That we don't actually know exactly what the back of Dunkleosteus looks like, which is pretty cool, interesting to think about. One thing I really like about the clade of placoderms that Dunkleosteus is in is they're so famous to be these big, scary predators all over the world's oceans. But in fact, they're not all big, scary predators. This is one of my favorite placoderms. This is an animal called Titanichthys. It's a huge animal, just like Dunkleosteus is, so something like 20-some feet long. So now Gnathostomes are getting quite big. But Titanichthys is like a filter feeder. It doesn't have any sharpness to its mouth, and we think it uses its gills to filter feed through like the plankton. So even in the Devonian, you're starting to you have these oceans, they're full of single-celled eukaryotes, they're full of everybody's little floating babies and everybody's little floating eggs that make up this big term called the plankton that obviously still exists today. And even back then, you have some of the biggest vertebrates, not as big, scary, bite you predators, but as big filter feeders on the plankton. That's a very ancient ecology. And here in the Devonian, we have animals like Titanichthys acting as these big filter feeders, which is really cool. So this animal and this animal are closely related. So you can think about them, very colloquially, like a basking shark and a great white shark are also pretty closely related. One's a filter feeder and one's a big, like, eat you up kind of animal. Very cool. More pretty placoderm pictures. Here's Bothrialepis. Bothrialepis is probably the most abundant placoderm. You can see there's its armored body right there. Here's this little visor. That little white spot there is where the eyeballs are. So both eyes way up front. I put Bothrialepis up here because it deserves to be up here. Bothrialepis is like probably one of the most abundant taxa in museum collections of any fossil. There are over 100, is it 120, Henry? Do you remember? How many species of Bothrialepis are there? Bothrialepis is known from every single continent. And what?
Approximately around there.
100-some species. It's a really interesting animal. My favorite thing about Bothrialepis, and when you think about this, when you saw that tree a minute ago and all that evolution happening in a relatively short time, there's a lot of experimentation happening. That's always going to be the case when there's a bunch of clades all showing up at about the same time. They're doing things similarly, they're inheriting similar anatomy, but then they're doing different things with that anatomy. Bothrialepis' armor completely covers its pectoral fins. And so its pectoral fins have on the outside of them armor and like a pointy spike thing. And so Bothrialepis is like often reconstructed, since it is an animal that lives on the bottom, as like walking around and doing stuff with like what look to me like crab legs. And they are a lot like crab legs because it's armor on the outside. Really cool, right? This is a vertebrate doing that with a bony tissue on its pectoral fins. Again, what's the... These are the things that I don't know where else to put them other than to tell you about them because I think they're incredible. Another relative of Bothrialepis is this animal called Microbrachius. Microbrachius is famous because it's the oldest example we have in the fossil record that everyone's very confident in of internal fertilization. So the reproductive behavior of all these early jawless fishes is really hard to know. You can assume maybe they're laying eggs behind a rock, maybe they're broadcast spawning, just throwing eggs and sperm into the water. There's at least one kind of placoderm that's doing this thing with internal fertilization. We interpret this to be a male and this to be a female, and parts of his armor near his pelvic fins are curved up. And so there's so many specimens of Microbrachius, you can see half of them have this structure and half of them don't. And there's things you're gonna learn about later when we talk about cartilaginous fishes and how they reproduce and how most of them have internal fertilization that lets us lead to the belief that some of these placoderms are experimenting with cool reproductive biology, having an internal fertilization of eggs. Doesn't mean she doesn't just lay those eggs right away, but the fertilization event is internal to her body cavity. That's very cool, very interesting, I hope you think. A lot of the really amazing information that we've gotten in the last 10, 15 years, well, maybe 15 years is more appropriate at this point, 15, 20 years, about placoderms comes from just one spot in Australia called Gogo. So out in the middle of the outback, actually like not, I mean, it's far because it's Australia, but not terribly far from like Ayers Rock, that big orange rock you might see when you watch a movie about Australia, is this reef called Gogo. These structures here in the outback are like the actual remains of the reef itself, like the limestone of like all the shells and the corally type things that made up the reef. And scattered all over the outback desert in this area are these rocks, nodules. And you break them open, you break them open, you break them open, and there's fish inside of them. And so I saw a talk about Gogo one time, and the paleontologist that worked there said, you bring up as many people as you can, and you all just break rocks all day, and you find one fish per day, per person, perhaps, is what they said. Which I thought was like, that's nice field work. It's like, you just sit there and breaking rocks with your friends and hanging out. But what's cool about these fossils coming from Gogo is that the preservation is incredible. It's three-dimensional, and everything's like, it's just the bones, but it's like perfect. So what they do is once they find a fish, they don't like chisel it out of the rock like we might do here at our museum. They dissolve the rock away because it's like a limestone. You can use like an acid to dissolve it away. This is a little placoderm from Gogo. There's this little face of this like almost 400 million year old fish, and all the little details are there of the little bones and the structure. So there's your eyeball, there's your little lower jaw. So McNamara aspis is a beautiful little fish, a relative of Dunkleosteus. One of the ways we know Dunkleosteus' body shape maybe is from little teeny versions of it, like McNamara aspis. So it's pretty incredible to me that we can see such detailed looks at like animals that are as old as this. Last thing to add on to placoderms on this slide anyway, is one of the most important animals that's ever been found at Gogo in Australia, is this fish here called Mater piscis attenboroughii. So it's named after David Attenborough, and Mater piscis means mother fish. This fossil has within it a preserved baby placoderm attached to like a yolk sac. So this is a female, she's got one little pup that she's obviously growing inside of herself with a placenta-like structure. So you guys are gonna learn that actually many, many fishes today do this. A lot of sharky things today do have internal fertilization, and then they do keep their babies inside and they give live birth. You might think that's a mammal thing. It is not just a mammal thing. But Mater piscis is this early placoderm that we have the fossil of it, and she's got a little tiny version of herself preserved inside of her abdominal cavity, which is really, really cool. So not only the earliest evidence of maternal fertilization, but also live birth in any vertebrate is a placoderm. Very cool. Okay, let's talk about placoderm anatomy. Placoderms have gone from being this like ancient armored group of fishes that people kind of think are interesting and then move on from, to actually being the hypothesis testing grounds of jaw evolution and a bunch of other evolutions of like early fishes. So I'm showing you guys this diagram here, and this is trying to show some evolution within the clade that Dunkleosteus is in between these like three forms. This one's from the early Devonian, the middle Devonian, and the late Devonian. And then there's a nice picture of the whole skeleton of the middle one here, this one. What I care about is not the evolution of something like Dunkleosteus. What I care about is you guys getting a handle on vertebrate tissues and where they come from. This is going to be a bit of like a bunch of information for you guys all at once to help you understand how incredible the evolution of jaws really is. So you can see this top line here, I don't know if you can read it exactly, but it says endoskeleton. Brain case, orange is jaw, cartilage, yellow is visceral skeleton, blue is paired fins and girdles. All these things, this maroon, these are all up there. These are all cartilaginous structures. So even in the jawless fishes we've already met, and now these early gnathostomes, the endoskeleton, the internal skeleton is there. It can rarely be found. And it's made out of cartilage. So there's a lot of familiar things in there to you. And then what placoderms do is have these really complicated bony pieces of armor. This is called the dermal skeleton. It comes out of the outer layers of their body. And so you can see pink is skull bones, and then there's some stuff divided up here. Cheek, operculum, which means the gill structure. Jaw bones is light blue, and there's trunk armor. I'm only giving you all this so you have something to kind of look back at once we talk. So there's an outer bony set of armor that's often found as a fossil, but we know from really well-preserved rare specimens that they have a whole cartilaginous endoskeleton that looks like this. So bone on the outside, cartilage on the inside, same elements as you might have in your body. Okay, that's some lead up and something you can look back on because we're about to get into jaws. Okay. Gonna turn the lights on just for 1 second so you can like shake your head. So what's gonna happen right now is I wanna give you guys some Roman numerals here, like 3 or 4 pieces of background information that I hope you can just like, okay, he's telling me a lot of crazy words. Okay, another set, okay, a different set. And all I'm trying to do is give you something you can look back on and understand the absolute craziness of how Jaws seem to have evolved. Because it's not fair at all for me to put a red tick mark and say Jaws. I could, and you guys might just accept that, but I can't. I have to tell you about how crazy it is. Okay. So the first thing, we're going back all the way to bilateria, the animals that have symmetry like this. You guys remember there's two kinds of bilaterians, deuterostomes and protostomes. Deuterostomes, when we're embryos, the first hole that forms the gastrulation is our anus, the second one is our mouth. What matters is that all bilaterians, you might remember this if you look back, you don't have to have it all in your head right now, are triploblasts. We have 3 cell layers as embryos. An ectoderm on the outside, a mesoderm in the middle that surrounds our body cavity where our organs are going to be, and an endoderm on the inside. And that endoderm is basically the lining of your gut, from your mouth to your anus or from anus to your mouth, depending on if you're a protostome or a deuterostome. These tissue layers are ancient, and what I care for you guys to know, because remember, we still don't really have very much bone yet in this class that's paleontology, except for this armor. We're about to get into bone, is where your skeleton really comes from. So you have here's your skeleton. You don't have everything I've listed here, but here's your skeleton. The ectoderm, when you guys were little triploblastic cells with 3 layers, from the outer cell layer, some of your bones form. Part of your skull, that's called your dermatocranium. It's basically all these bones that you can actually see on someone's skull, their lower jaw, their face, the top of their head. That's the dermatocranium. Other animals that have armor, like armadillos or armored dinosaurs, that's the layer that this comes from. You have a clavicle. This bone right here, only this one off side of your head, is from your ectoderm. None of your other bones below your shoulder are from your ectoderm, but this one is. And then things like your kneecap. Don't worry about it for now. That middle layer, the mesoderm, is that like contained middle cell layer. From there is where we get our neurocranium. The neurocranium is the brain skull, the nerve skull. When I gave you for vertebrata skull as a character, a ctenophore for all vertebrates, what I really meant is a neurocranium, a cartilaginous safe housing for all those nerves that make up your brain. So your neurocranium is from this cell layer called the mesoderm. The mesoderm is also where all your vertebrae, all your ribs, all the rest of your arms and legs, almost your whole skeleton, except for a little bit of your head, and this bone right here, comes from a different cell layer than your skull and this bone. I know that's kind of cool and interesting, maybe. There's also a thing called the endoderm, that inner tissue layer that lines your gut. There's only a handful of bony bits that come from your endoderm. It's called the gill skeleton. Here's this terrible word, I only say this one time and I'll never say it again. Splanchnocrinium is technically what it's called. You can think of it as the gill skeleton. For you guys, that's not so relevant, but you're about to see it's really important. Okay, so we know all these things to be true. We can watch embryos develop like this and see parts of the skull, parts of the body come from different tissue layers. They do not form in the same way. That's observable. How does it map onto the fossil record? How can we take other animals and learn more about ourselves? All right, that's that first slide. Here's how that might manifest itself. Here is a shark skeleton today made out of cartilage. Here's a bony, fishy actinopterygian skull today. Here's a bony sarcopterygian skull today. It's a lobefin fish that you guys would probably call a chicken, but it still is a tetrapod thing. And so there's a lot of colors here. These colors are shared intentionally. So I'm just trying to blow your brain a little bit. So that ancient vertebrate skull, the neurocranium, the skull that unites all vertebrata, is cartilaginous. It comes from the mesoderm and then it forms into bone. It surrounds your brain. In a shark, it does not form into bone. This whole thing, this whole shark skull is blue. That's the neurocranium derived from the mesoderm. We can only see it in this fish here and here. I know it says orbit there, don't worry about that. This is the good place you can see it on this fish. On the chicken, you can see it. It's all the actual bones, and there's many of them, that surround the brain and part of the ear. There's also a little bit up here in the bird. So inside, deep inside that skull of that chicken is that mesoderm-derived neurocranium. The gill skeleton, which actually has a lot of really cool components, are colored green, purple, and brown here. Remember, if we take a lamprey or something, we can see those same gill arches. Or if we take another deuterostome that has those pharyngeal gills, we can trace the arches. So this shark jaw is the first gill arch in like a lamprey or a hagfish. This is the second gill arch in a hagfish or lamprey. And then normal gills. So there's our green stuff, and there's our purple stuff. All I'm trying to do is show you that parts of these skeletons are homologous. If you guys ever Google like a bird sticking its tongue out, the bone inside of a bird's tongue is deeply homologous with the second gill arch of a shark, the second gill arch of a lamprey, these bones holding up the gill structure of a fish. Super weird, it's super crazy. These are the kind of things that people knew in the 1800s and were like trying to figure out what to do with. Okay, I know it's a lot. The last thing we're putting up here is that outer tissue layer, the dermatocranium, the bony dermal bones. You can see there's only red in the osteichthyons. Most of the bones that you would recognize as a bird skull, most of the bones, if you ever catch a fish that you're seeing on the outside of a bony rayfin fish's skull, are from the ectoderm, the dermatocranium. Living sharks do not have anything like a dermatocranium. Okay, lots of different words up there. Second thing to talk about, how bone forms. We're going to talk more about this on Thursday. There's two kinds of bone in your body if we break it down to its simplest levels. One is intramembranous, which is ectodermal bone. What happens here is in that outer tissue layer, there's these progenitor little cells. They're called mesenchymal cells if you take evolution or if you take developmental biology. They lay down like a lattice of structure. And then these cute little cells called osteoblasts come in and they lay down bone tissue. They lay down calcium carbonate. And as they do it, some of them stay behind and become these cells called osteocytes, so cells that live in the bone. Bone is a living tissue. This thing kind of grows like a sponge grows. That's dermal bone. That's this bone in your head. That's your clavicle. The other way bone forms in your body is called endochondral bone. It's extremely different. In your little developing embryo, a lot of these little cells that make cartilage form a little shape. They form a shape out of cartilage. So this is already happening in things like placoderms. And then what happens is they invite in, through blood vessels and things like that, bony cells that start to lay down bone. These little cells called osteoblasts. They lay down the calcium carbonate. They leave little tracks for nerves and blood vessels to follow. And as that bone grows, it takes over the cartilage. The cartilage does it first, and then the bone follows. So endochondral bone is internal bone for you that is first laid down in cartilage. All your mesoderm bones and all your endoderm bones are formed in this way. Again, this is a lot of information. I'm very aware of that. And it's just trying to give you guys a little bit of these terms. Intramembranous bones, which form the ectoderm, endochondral bones, which are preformed in cartilage. Here is a hilarious cartoon that new moms and dads get when they go to the hospital. It's obviously just like an upside down femur, an upside down femur, another femur, another femur. It's just a cartoon. But I saw this and I laughed because I thought this would be so helpful for my classes. So when you have a new baby, some things are happening in its bones inside of its body, where in the middle, there's bones starting to grow and it's growing up and it's growing down on the femur and on the tibia. And that's why babies have squishy hands and feet. The cartilage is still right there. But the bones in their head are very different. They're starting from the middle and spreading out in kind of this spongy texture. So in this baby human, with this like piece of info you get at the hospital once you have a baby, you can actually see, I hope, a little placoderm with a cartilaginous skeleton, pectoral fins, pelvic fins, and vertebrae, and then armor at the head and the gill structure. That's the clavicle and the skull. So just like a placoderm has armor all over its head, a little bit of armor at the top of its fins, and the whole skeleton is cartilaginous, you can kind of see that here if you squint on us, you as a little baby. It's very cool. So I want to talk a little bit about how these tissues form. Cartilage is super, super, super ancient. All the fishes we've met so far have cartilage making up the hard tissue of their bodies. Here's a shark, that vertebrate skull, shark neurocranium, it's all made out of cartilage. You guys don't have so much cartilage in your bodies. This. Ends of your joints are cartilaginous. As people get older, the ends of their cartilage joints finally become bone. That's when they get all rigid and it hurts to walk and stuff, right? Finally, these things happen. So you don't have to know all this stuff. I just think it's interesting for you guys to think about. Cartilage is super ancient. And in fact, other things that are related to cartilage, like the collagen proteins of cartilage, are like shared by things like arthropods and stuff. So this is very ancient animal tissue, all vertebrates inherited. It's mostly collagen, it's mostly protein, that's why it's squishy. It doesn't have any mineral component. We won't spend too much time on these. Our endochondral bone, like I said, has a cartilaginous precursor. Cartilage is easily the oldest of the tissues. So the other thing that we're going to talk about is dentine is a bony vertebrate tissue. Dentine is what's in your teeth. The white part of your teeth is chemically really similar to bone. The difference between bone, which proper bone we're going to talk about, endochondral bone, we're going to talk about on Thursday. Difference between dentine, which is really like bone, and then the bone like in your arm, is that there's like fewer cells that are alive inside your tooth. Your dentine can get eroded, like when you have a cavity, and it doesn't heal itself very well at all. A bone heals itself if it breaks. So dentine's a little bit different. I'm only telling you this because if we look at these ancient fishes, these agnathans you guys have already met, their armor is made up of bone and it's covered with all these little teeny things called odontodes that are made of dentin. So you have this structure in your body right now. These ancient fishes you've already met have intramembranous dermal bone armor, and then that armor is covered with these little teethy things. I've already kind of hinted at this. So that little bump on this fish has dentin. On the outside of that dentin is enamel, and there's a pulp cavity on the inside. A lot of the details are shared. We can look at these ancient fishes, and people knew 100 years ago these things have tooth cell or tooth tissue type on their armor. Something that I think is kind of fun for you guys to think about is like mammals do a lot playing around with these tissues. We're going to talk about that later. This is obviously quite a big tooth. Elephants don't have any enamel on their tusks. It's only dentin and it's ever growing. We're going to talk about that later. Again, these tissues, dentin is only ever found on the outside of a body, and it's only ever coming from ectoderm. Last tissue I want to talk about is enamel. Enamel, the difference is it's laid down just like bone is, it's laid down just like dentin is in terms of cells laying it down, but the difference is that it's almost all crystalline structure. There's no cells in the matrix of enamel. And enamel's only on the outside of your tooth and on the outside of these guys like scaly, bumpy things on the outside of shark's armor. Enamel is mineralized. It's not a living tissue. It's very hard. It can do a lot of cool things. But when you get a cavity and your enamel erodes, unfix it. Your crystalline structure is gone. So the dentist has to give you a filling. There is no healing your tooth like you heal a broken arm. Okay. Enamel on the outside of this fish's armor, enamel obviously on your tooth. And what's cool again, mammals are going to play with these tissues later. So here's how like a rodent, like this beaver, might take this difference between how enamel erodes and how dentin erodes to like evolve a bunch of really cool teeth. We're not going to talk about that now. Again, only from ectoderm, only on the outside of your body. Last thing I'll say, I've already hinted at it, this is number 4, hard teeth and scales. I've referenced this last week. There are these deep homologies people have noticed for a long time that have prompted people who study early vertebrates to wonder, how do you take these obviously skin structures, and on sharks we still see the same thing, and turn them into teeth? What is this relationship between these dermal scales on the armor or loose on a shark and the teeth? So again, all of these have the same deep architecture, a living pulp cavity with blood vessels and nerves, a dentin layer, and an enamel layer where it's actually sharp and pointy and does interesting things. So this is the cross-section of a shark's scale. This is a cross-section of a shark's tooth. This is a cross-section of your tooth. All these 4 big pieces of information, developmental biology, how bone forms, these different tissues, how scales and teeth seem to be related when you put them under a histological slide. These are all bits and pieces of this story. And I'm going to try to bring it together for you now. So let's talk about how jaws must have evolved. Here's our nice shark skeleton. You saw this exact slide when I gave you that jaws synapomorphy. So here's this animal with giant dermal bones and no teeth. Here's this animal alive today, giant cartilaginous face and gills. Here's this bony fish today, bones full of teeth. I think we all would say they all 3 have jaws, but these jaws are extremely different architectures. So what's the deal? Let's go through it. This is our best working idea of how jaws must have evolved. We can then use this as a framework to like test hypotheses. So here's our jawless fish skeleton, like today a lamprey. We can see this. There's a cartilaginous skull, that's that vertebrate skull, and there's cartilaginous jaw arches, tons of holes. If you look at a lamprey, there's a bunch of gill openings. Okay, all made out of cartilage. In these early placoderms, what we can see is that first jaw arch, or gill arch, I'm sorry, that first gill arch basically becomes the jaws. To me, it's really interesting to imagine these tiny, you can imagine how natural selection might work. If you can start to like flex that front gill, you can start to do things maybe. I know that sounds really hand wavy, but it's for me really fun to imagine because it's hard to get these teeny little fossils, especially with their cartilage preserved. But that first gill arch we know is homologous with the actual upper and lower jaws of things like sharks and you. And by the time we get to things like sharks, that second gill arch has joined. Sharks today have this complicated, heavily reinforced jaw structure, but these 4 pieces of cartilage in a big shark's mouth are homologous with 1 and 2 gill arches in like that lamprey. Really interesting. There's teeth in there though. So there's those 2 gill arches in a shark, but a shark has teeth on the cartilage. Again, one thing that's really cool about sharks and different from us bony vertebrates is that because it's all cartilaginous, there's only a couple points where these things actually touch each other and hook up. So sharks have amazing mobility in their skulls. Just because I've been saying stuff at you for a long time, I'm gonna let you watch this for a second. Forget that, there you go. So look at the jaws and look at the gills, how they're related in that goblin shark. So that feature, which has a very specific name that we'll talk about later, of the articulation between these fancy cartilages, which we're going to call the palato-quadrate and Meckel's cartilage, the upper and lower jaws of sharks, that mobility is not like necessarily ancient. Modern sharks have evolved that, but it helps you see. It's kind of like he has all these teeth and it's like this poor fish is even getting chewed. It's just getting engulfed. Okay, so this might help us understand shark evolution, right? Cartilage, cartilage, cartilage. This is why this is such a problem. Armored and bony gnathostomes, you don't have a cartilage lower jaw, and neither does Dunkleosteus. It's bone. You have teeth, Dunkleosteus doesn't have teeth, but you both have bone. So the question is, how do we reconcile this? Talk to your neighbors for a second. What was this synapomorphy? You can look back. Anybody? Anybody? Yeah, that's the dermal skeleton. So the bony armor skeleton. Now, up here in gnathostomes, that's where the placoderms are, that's where the sharks are, that's where you are. So something's going on. The ancient inherited feature, though, is these giant bony plates everywhere. Okay. This is hilarious to me, and you guys are going to talk to each other. I'll walk you through it. This is a phylogeny. Everything that's green in the phylogeny is one of these placoderm armored fishes with jaws. Here's one jawless fish right here, being cute. Here's one bony fish. Doesn't matter what it is, but it's related to us, bony fish. And what you're seeing, there's cute pictures of them. What you're seeing is their lower jaws in cross section. The purple is the Meckel's cartilage. So the actual cartilage part that a shark has, these are the homologous bits of that cartilage in all of these fishes. And then the brown, which I don't like all these colors mixed around, I'm sorry, I didn't make these figures, you can blame these authors. But the brown there is the dermal bone, the armor bone, sometimes with teeth, sometimes not with teeth. What I would like you guys to do is look at this for a while, how that cartilage, which is homologous with a shark's cartilage, how that bone, some of which is homologous with your bone, and sometimes with teeth, make some observations, talk to your neighbors. You see any patterns? You see anything you want to talk about?
Yeah, sort of like the inner outside. I wonder if it like is there you find this job together? Maybe.
Well, like the other is that. Those are funny. Multiple points of holding. Yeah. It's funny, it's like closest to the two kids, T-plus, and like it's especially in the older. It's not good. It's like the version of
No, I'm not even clear.
I'm going to let this like several seconds.
All right, I don't need anybody to be super fancy and try to interpret this whole thing. What are some things you're just observing at all? Somebody hasn't said anything yet.
The dermal bone moves around a lot.
The dermal bone moves around a lot. These are all lower jaws, right? It's not like this. Sometimes it's top, sometimes bottom. These are all lower jaws. Okay, so dermal bone moves around a lot. What else?
Teeth appear in this sphere multiple times.
Yeah, if these things are all teeth, teeth are there, not there, arranged a certain way, arranged a different way, the bones arranged a different way. I wanted to show you guys this because I know I basically like tired myself out and used up all your patience by saying everything I just said about embryology and tissue structures and all this kind of stuff. And what's so great to me is you can come in with all this prior knowledge. This paper is pretty recent, right? It's 3 years old. This is the actual state of what we know about the real anatomy of placoderms, and it's a nightmare. Obviously, there's tons of experimentation happening. Once you get cartilaginous jaws, once somebody's got their little cute front gill arch as like a movable thing, a lot is happening with bone, a lot is happening with teeth. Those teeth are obviously coming in from the odontode little bits that are on the armor. It's not a simple story. It would be great in like a college class or a high school class to teach the evolution of jaws and be like this, which is like kind of what that first figure I showed you did, if you want to explain what a shark is, but that's not what we really see. It's crazy. I don't think I can tell you what the pattern is here. You guys saw how those fishes all kind of show up at the same time, and for tens of millions of years they live together. There's obviously a lot of experimenting here. Your cartilaginous skeleton, your dermal intramembranous bone skeleton, your teeth that are sitting on top of that actually become the things we call teeth. It's not clear. Isn't that unsatisfying? Anyway, here's why it's a problem. People have, for 150 years or more, have talked about sharks. Sharks is a good example. They have a cartilage skeleton. They're the ones to compare us against. But now that we have all these fossils, it's like, no, it's not that simple. So here's McNamara aspis, who you met from GoGo. Here's a couple other placoderms. And now here's the crown of Gnathostomata. So here's some other bony fishes. There's you as a bony fish. Those colors are real. Some of these placoderms have this bone, which is the jugal. It's this. Some of them have this bone, which is a maxilla. It's this. They have these bones. We can track that ossification all through evolution. Well, guess what? What the heck are these sharks doing? Sharks, as we understand them today, modern cartilaginous fishes, must have lost dermal bone and started to do their own thing during all this experimentation. It doesn't make sense to be like lampreys, sharks, fishes like us. Because we know all this stuff about placoderms, we now know this thing is sitting in the middle there and it sucks. And nobody knows what to do. Do you think that's fun? I think that's fun. This is like maybe the nittiest, grittiest we'll do for a while now. But I just had to give it to you. So one thing that I think is really cool that we can put on our tree is somewhere within placoderms. Here's the placoderms. Some of them yes, some of them no. We can put a tick mark, a synapomorphy that goes here, and that synapomorphy is the homologous things in your mouth and a shark's mouth that are called teeth. So you've now seen a diagram like this a bunch of times for me on different days, how a shark's placoid scale and a shark's tooth both develop in the same way and have the same tissue structures. But teeth, and this is just how your teeth form too, are a deep thing that some placoderms have and everybody after them has them. So synapomorphies for teeth goes kind of right there in the middle of placoderms. Okay. Let's take like a 3-minute break because I know that was kind of a nightmare. I'll ask you guys for your thoughts on that because that was my first time I ever tried to give it all at once and maybe you hated it. Okay, take a second. All right, so now we have our jaw vertebrates. I hope you guys now are kind of sick and you just would have trusted me that I said jaws evolve here and now I've tried to really cram it down your throat how crazy that is. And now we have our teeth. Okay, let's keep going. This next little grade of biodiversity are these animals called acanthodians. So acanth is like a spine. Whoops. So here's acanthodians. They evolved in the Silurian. They're some of the earliest fossils of jawed vertebrates we have, and they go all the way through the Permian. So they live for quite a long time. Some lineages make it past the Devonian just fine. These have often been called spiny sharks for a long time. You can see they have some very different body plans, and they're really, really, really spiky. Some of them have like whole lines of spikes along where those early folds were, you know, between pectoral and pelvic fins. So these fins and these fins, they still have other ones that are spiky. And so what's cool, some of the ones that survive into the periods after the Devonian, the Carboniferous and the Permian, a lot of them are these filter feeders. So they're kind of living like an anchovy lifestyle. They're little guys, probably out there in swarms, schools, filter feeding, which is really interesting. These guys are found in fresh water often, which I think is very interesting. So if you were to go fishing back in the Paleozoic, in early lakes and streams that have some plants around them finally, things like acanthodians are a lot of the fishes in those freshwater systems. The thing that's cool is on every single one of their fins, pelvic fins, pectoral fins, dorsal fins, which we talked about, anal fins, which we haven't talked about yet, everything except for the tail, basically, there's a spine on the leading edge of it. So these are spiky, spiky fishes, which are pretty cool. They are also really complicated. You saw in the phylogeny, some of them, maybe all of them, but at least some of them are actually with chondrichthyans within our crown group, jawed vertebrates. Other ones probably are outside of the crown. So this is a grade of organisms. Acanthodians probably let us know what the like ancient crown group Gnathostome common ancestor looks like. The Luca of Gnathostomes is probably something, right? Something like an acanthodian. They have bone, they have cartilage, they've got proper jaws with teeth in them. And so the fact that some of them are closer to sharks with dermal bone with a tooth in it is really interesting. If these sharks we now know today don't have any of that dermal bone, they have their cartilaginous understructure with tooth on it. So that's acanthodians, just so you know about them. So here they are, a couple of different groups, one of which, maybe more than one of which, is actually closer to things like today's cartilaginous fishes. All right, now let's get into those. So if we go within gnathostomes, we have the two big groups today, cartilaginous fishes and bony fishes. We'll save bony fishes for Thursday. Today we can talk a little bit about cartilaginous fishes. These fishes are often the placeholders in your brain for what primitive fish look like, but I hope you can see it's way cooler than that. So for chondrichthyes, cartilaginous fishes, here's the two groups that are alive today. I have some snapemorphies for you guys. Two of them for chondrichthyans that are true back in the Devonian and the Silurian, and they're true today. The first one is they have a completely different kind of cartilage than everybody else has. The cartilage in your nose and the cartilage in your ear isn't really mineralized. All chondrichthyans, like this stingray and this shark, have what's called prismatic calcified cartilage. They do put calcium structures into the cartilage of their bones that makes them really rigid and tough. It's kind of formed as these little tesserae, little hexagons, pentagons, heptagons that all fit together. So under a microscope, you can always tell if a chunk of cartilage belongs to a chondrichthyan or if it's just a chunk of someone else's cartilage because of this structure. So this is a living shark, of course, skeleton. The only part of this that's likely to make a fossil are those hard teeth that have dentin and enamel. The rest of it isn't. But shark bones do stay, shark skeletal bits do stay around for quite a bit because they are prismatic calcified cartilage. You can find them on a beach, washed up. A lot of times when sharks die out at sea, and their bodies fall apart and they wash up on a beach, it looks like a big snake monster because all the soft stuff's gone and there's this weird train of vertebrae. So you can look that up if you want to. But prismatic calcified cartilage is a chondrichthyan snapemorph. It's a special kind of mineralized cartilage that only they have. The other feature that unites all chondrichthyans is very interesting. It's called claspers. So on the pelvic fins of these fishes, so you have your pectoral fins up front, your pelvic fins in back, the males, and only the males, have these trailing structures called claspers. These animals have these things and they use them as what are called intromittent organs, analogous to a penis in like any kind of land-living vertebrate. So almost every single solitary living chondrichthyan has internal fertilization. Whether they lay eggs or do anything after that, the actual fertilization of the eggs is internal. So there's a male and female of this little cute skate today. And here's a couple different sharks. When sharks, and stingrays, but it's easier to see with sharks, when they mate, the males grab onto a pectoral fin of the female, and then he curls one of his claspers, left or right, depending on what side he's on, and then he has his sperm released, and it follows down a groove on the fin to enter her body. Female sharks have like extra thick scales on their pectoral fins, because when they mate, they have to like get bit by this dude and hang on like that. Not something people think about all the time, and I think it's something that can be surprising to people or students, especially if you haven't ever learned it before, that the default condition for cartilaginous fishes isn't spraying eggs and sperm in the water, it's internal fertilization. So those claspers are very ancient. So here's chondrichthyans and how we see them in the fossil record. They go all the way back to the Silurian. We have loose bits of scale, these special placoid scales that we can recognize as chondrichthyans. Of course, things like sharks and stingrays today are chondrichthyans, and then these other animals called chimeras or ratfish, which we'll talk about later. What's cool is that divergence between the two living groups absolutely takes place way back here in the Devonian. So both those lineages that are on that previous slide are very old. As a rule, chondrichthyans are carnivores and they live in the oceans. We're gonna have multiple times, especially during dinosaur times, when there's lots of freshwater chondrichthyans. But for the most part, they're marine and all that. They kind of have this classic early fish body plan. You guys are gonna see that and be like, it's a shark. Look how old sharks are, and it's true. Versions of this clade keep a body plan like this all the way up till today, which is really interesting. A lot of our earliest fossils from the Devonian are pectoral and pelvic fin girdles, if it's not jaws and teeth. And you can see there's spines that lead the edges of those fins. That's not true in most sharks and rays today, but in the earliest members, just like in acanthodians, there's spikes at the front of all their fins. Kind of cool. So that's Doliodus. One of the best known early chondrichthyans is an animal that lives alongside Dunkleosteus in those rocks from Ohio. You can find it in other places too. If you guys Google image Dunkleosteus, that big scary placoderm, 9 times out of 10, the thing it's biting in half is Gnathostomata, this early shark relative. So you can see there's these big spines at the front of the fins. Look how big these nice big pectoral fins are. There's other features we could talk about for these guys, but there's early sharky shark looking things that are true chondrichthyans all the way back this long ago. There's a nice reconstruction of Gnathostomata. So you're getting familiar gnathostomes. All right, on the other side of the tree, we've got our osteichthyans, our bony fishes. And we're gonna talk about those on Thursday. And once we get to them on Thursday, I'll give you 3 snake markers for them. But if I remember right, that's it. So I'll see you guys on Thursday. And we'll talk about us, bony fishes. All right, thank you.